Okay, so I ordered a CRISPR kit online a few days ago. It's the Odin CRISPR kit. If you just search up Odin CRISPR kit, you'll find it. It was a really great way to get some hands-on experience. I decided to document my experience with it, so here's how it turned out. So to start off with, I'm just going to explain what the purpose of this is. So CRISPR is a gene editing tool, which I'll explain more in depth in another video. So this kit allows you to modify a strain of E. coli to be resistant to streptomycin, which is an antibiotic that normally binds the ribosome of bacteria and prevents it from making proteins, which then stops it from replicating and growing. So this kit makes a mutation in the ribosomal subunit protein RPSL that prevents streptomycin from binding it, which allows the E. coli to grow on the media with streptomycin, which is our desired end result with this kit. First, I had to make agar plates, which is a pretty cool experience. So basically, they sent the materials to make agar, which is that yellow gel you see in the petri dishes. So I just poured that in and waited a few hours for it to cool. And so it's a growth medium for bacteria, so it has nutrients that support their growth. And once the agar plates were done, I just added some sterile water to the E. coli DNA that was sent, 100 microliters to be exact, just to rehydrate the DNA since it was freeze-dried, so adding water made it actually usable for the experiment, so I could actually pipette it and spread it around. And then after I added the water, I just shook it up to make sure it all mixed together. Okay, and then after that, I took one of the agar plates I had made, which, I mean, look at that. I was really proud. It kind of looked like it would in an actual lab. So yeah, I took the DNA that I had just added the water to, and then I pipetted it all onto the agar plate. And then after I made sure to get all of the DNA on the plate, I just took a little inoculation loop and gently streaked the bacterial DNA all across the agar plate. It was actually a really therapeutic experience. And then after that, I just put the lid on, flipped it upside down, and waited for about 3 days for the bacteria to grow. Just put it out on a table, room temperature. And then successfully, it did grow. Look at all that bacteria. And now I'm going to mix the bacteria with 100 microliters of transformation mix, which basically what it does is it helps the plasmids, which are like little loops of DNA in bacteria that carry genes, that I'm going to add later on, penetrate through the bacterial cell walls so the bacteria will actually pick up the plasmids. So basically, the mix weakens the cell wall and makes it easier for the bacteria to pick up the DNA that we want it to. I'm just going to take an inoculation loop and scoop up some of the bacteria. And then I'm just going to mix it in with the transformation mix. And then using a micropipette, I just kind of gently pipetted it up and down a few times to make sure it really mixed in. And then finally it did all come together really nicely. And then here I'm adding 10 microliters of the Cas9 plasmid, the gRNA plasmid, which contain instructions for making them, and the template DNA that contains the desired mutation to the bacteria. So the bacteria is going to treat the plasmid DNA like its own, so it's going to produce the Cas9 and the gRNA. And so what's going to happen is basically the Cas9 protein cuts DNA, and the guide RNA attaches to the Cas9 and gives it instructions for where exactly to cut, since its bases are complementary to those of the gene that we want to edit. So once the gene has been cut, the bacterial cell is going to repair the break using the template DNA that we've provided, which is almost identical to the original DNA except for a single base pair change. And that one change leads to an amino acid change during translation. So a lysine is going to be swapped for a threonine, and that's what's going to alter the structure of the RPSL protein so that streptomycin can't bind to it. After that, I incubated the edited bacteria on ice for about 30 minutes, and then after I did that, I incubated it in warm water, which is shown here, for about 30 seconds. And so what that does, cooling the bacteria and then heat shocking it, is increase the efficiency of transformation, which is the term for when bacteria picks up foreign DNA, like the plasmids that I added. So the ice incubation helps the cell membrane stabilize more, since remember the transformation mix kind of loosened it, and it prepares it to bind the DNA. And then heat shocking it in warm water temporarily loosens the membrane, which creates tiny little openings that allows the DNA that we want to slip inside before the membrane seals up again. Because remember, it doesn't want to be too permeable. It doesn't want to just let everything in. So here I'm taking a tube of powdered luria broth. And luria broth, remember before when I made the agar plates, those are made basically by adding agar, that gel, to luria broth. So it's the same nutrients that are in the plates that help the bacteria grow. And so here I'm just adding some more sterile water to it, about 250 microliters. So just rehydrating it so that it's like liquid and it can actually be stirred in with the bacteria. And after that, I incubated the tubibacteria for about 12 hours at room temperature. And so I didn't show this on camera, but I made more of the agar plates. Oh, look at that. This time the plates have streptomycin added to them, to the agar. So now I'm adding the edited bacteria that I let incubate for about 12 hours onto the plates. And so if the experiment worked, then when I check back on the bacteria in about 3 days, there should be signs of growth, since it should have the gene for streptomycin resistance now. So yeah. So yeah, now I'm just taking an inoculation loop and just streaking the bacteria all over the plate. That's how it looks right now, so hopefully it'll grow more when I check back. And I'm just going to turn the plate over, let it sit there for about 3 days. Before I can just completely leave it in peace, I have to make a control group. So I'm adding some of the original unedited E. coli that we grew at the beginning back onto the plates with a different plate this time with streptomycin, just to serve as a basis of comparison for growth when I check back on the results, so I can make sure that any growth that is on the plates is because of the CRISPR, because of the plasmids we added, not just totally random. So yeah, hopefully nothing will show up on the control plate. It worked. So as you can see, the experimental group, the one where the bacteria were edited, you can see there's growth on the agar plate. So you can see all those colonies have spread out and they've grown. Whereas the control group, where the bacteria weren't edited, the only bacteria on there are the ones that were originally streaked. So there's absolutely no growth. This was a really great experience. I've been obsessed with CRISPR for about 2 years now, so actually getting to use it hands-on was a rewarding experience. I'm definitely excited to keep pursuing this in the future when I go to college and study genetics. So yeah, and I would definitely recommend this kit to anyone who's interested in learning more about CRISPR. So thanks for watching.